All right, so welcome back for the lecture, the plenary lecture of by Toshio Fukuda. And I think it's really a great honor to have Toshio here with us today. I mean, he has been, as a roboticist, involved in this whole field of biomimetic robotics for a long time. And I think one of the outstanding examples of his work is the attempt to build a robot based on the monkey. And this work started in 1985 and has been summarized here in a book that was published very recently. So this is here about 25 years of hard work trying to understand biomechanics of monkey behavior. And you might have all seen these videos of the swinging robot monkey. But Toshio is director of the Center for Micro and Nano Mechatronics and a professor at the Department of Micro and Nanosystems at Nagoya University. And he also has a position at Seoul National University in the domain of robotics and microelectronics. And if you read his bio, it's an impressive list of honors and important positions. So in that sense, I think we have a lot to learn, and I'm really looking forward to your presentation. It's great that you want to join us. So thank you.
Thank you very much. Good. Thank you very much for introduction. Yes. So today I'm going to talk about micro, nano, biomimetic, and biohybrid devices that Tony asked me to do something. Okay. So before going to micro, nano, there's a I'm from here, Nagoya, in Japan here. Okay, Nagoya here. So here, so Fukushima, so there's some nuclear power plant here, bomb out, but it's about 700 kilometers away, so nothing to me. Anyway, but Ishiguro Sensei, who is here somewhere, he's from Sendai here, so he's affected by earthquake. Yeah. So anyway, but he's originally from Nagoya, so. So anyway, this is a girl here, and we are in here, Barcelona here, okay? So that's a nice thing. First of all, before going like a micro, nano, those things, I'd like to talk about some biomimetic robot system. One example, like a brachiation robot or a monkey type of robot. Just he showed the books like that. Also, like maybe I can talk about the background of cellular robot system, CBOT. Cellular robot system here, so like some work about over 20 years. So then micro nano bio devices, this is today's main topics. And then I spent 2D, 3D cell assembly system, and then I'm going to signal cell analysis and characterization to make 2D, 3D cell assembly system. And then maybe single cell adhesion measurement, or cell adhesion, or cell cutting, or whatever it is, and others. Then go and talk about that. First of all, living machine. So, like Tony like it, okay? So, living machine. So, it's a kind of, from my viewpoint, this is a kind of multi-scale system from nano, micro to a macro level here. When you're talking about such a thing, energy varies also can think, also is maybe also nice for energy saving, also environment saving, something like that. But we can talk many things, okay? And then when we talk about robot system here, then, well, the reason why before that, there are so many kind of conferences still going on, like artificial life conference, or kind of adaptive behavior conferences, everything going on, starting from somewhere in UK. So that's why, okay? So, like, so then when we talk about robot system, we can talk about kind of biorobotics. Biorobotics also kind of includes such a bio-inspired robotics, biomimetic robotics, biosynthetic robot, biohybrid robotics. But the thing yesterday I mentioned, there's a panel here. I just want to have this kind of thing to understand such a nature, biological system, discover some new mechanism for something like that, then synthesized, and then designed. So this is a kind of way of from science to engineering, back to science engineering, like that. Such a kind, we can make a kind of feedback. So this is today, first I talk this part of things. This is more like a macro level, okay? Then we go more here like that. Why I'd like to say why I'm interested in these areas. That's a kind of question. So in my lab, we have those things here. So like a monkey type of robot here, which I started in 1985 or something like that. So that's a thing. So I like this one, and then later on I made this one. Then later on, also last time I work with such a kind of a more robot. Why many robots work together? How do they do that? Like that. It's a it's also 1987. I wrote a paper on such a self-reconfigurable robot system, like a kind of a Lego type of robot those days. Okay, so then again went to some here like such a MEMS type of micro areas, nano robot system, and spin out is a kind of medical robot. So that's the thing. So that's a kind of thing. So maybe those things can be maybe interesting. Some people already know, but I knew some people who are not in the robotic field, maybe better to show something like that. Where I used to work, so it's kind of about more than 20 years ago. So you're moving here, right? Even if you change here, okay, the distance, they can adapt by themselves. So this is kind of here, this is kind of color ball is a kind of trick because we measure the kind XYZ position, okay? So that's a kind of feedback over there. So this is a kind of monkey. So that's a kind of reinforced learning scheme, what it should be, like that. That's a kind of a question those days. So now then I made a kind of similar one. So like one robot, okay, working like that. So, well, this nowadays many biped robot work is there. So not so, but this is a passive walk, half passive walk. So this robot can walk 5 times longer than active robot using a ZMP policy, okay, with the same energy sources. That's a kind of thing. So you can see because of that here the robot, knees always stretch out. ZMP type of robot always bending, okay, to avoid singular point control. So that's a. So then the same robot, this is the same robot, okay? Same robot, okay? So this distance is not equal, okay? But there's a thing, long, wide, small, like that. So this even it's not equal, so they can move around. This is with a backswing, like that. So energy kind of control in a way. So the long distance like that. That's that. So also like this is kind of biped, okay. Then this is a quad, 4 leg, okay. If they try to move such a slope, slipping, but still this is only 20 degrees. But biped cannot walk, so need a kind of change of locomotion mode from biped to quad. So quad, there are many kind of walking. Say like kind of crawl motion, 1 leg out of 4 use, or 2 leg out of 4, even 2 out of a leg. There's a kind of such a pace motion or trot motion. Then energy-wise, speed up, then go to the gallop. So it's a kind of like a gear shift of the curve. So that first when slow, then just a crawl motion, then go to the second gear, such a kind of pace motion. Then third gear, that's a trot motion. Fourth gear, just a gallop. That's a kind of locomotion. Then we can assemble those in everything. So this walking here, come up here. So climb up the kind of still kind of ladders. So ladder up here, nothing special. This is kind of flat. Okay, so nothing to keep. That's flat. Okay, so if you're not good, so it's a just kind of physically, kind of physics. Okay, so climb up here. Only up is just like this. Okay, that's all. So the climb up. This is like a kind of pace motion, like a camel moving this way or this way. So now the robot. Just wait a minute. 1 minute. Just kind of start with the problem. Don't fall down. Okay. Okay. There's some water. Just a kind of little freedom of degree of freedom is just less than the human. So that needs something trick here, so that if they can start moving here, or maybe here's a bananas here to catch a bananas. Okay, okay. So something like that. This is a kind of a thing. So you can see this is a kind of sort of biomimetic monkey base. Okay, so that's a kind of biomimetic robotic system. Then you can say many robots work here, who can, as I said, this can something like that. So also like here, this is a simulator of human blood simulator, human being, particularly for everybody. Everybody have a different type of kind of blood vessel pattern. So it's a patient-specific kind of vessel, and somewhere somebody have some kind of aneurysm here inside the brain, and then need a kind of simulation before the surgery. Maybe this is a kind of also look like a living here. So here this is kind of made by silicone. Here, so look here exactly. This is just a 10 micrometer technology. Then you can make it here. So kind of platinum coil inside, fit in, so that the blood will not flow inside, and to prevent the kind of aneurysm will grow and blow it and die. Okay, that's something like that. So that's a kind of thing here. So this is a kind of. So this is a kind of sort of thing, sort of like a human biomimetic here already here. So also like here in this way. So now I wrote a book on kind of material locomotion robot here. Just he explains that here, and then why I showing this one? Because DARPA challenge. I'm not challenging the DARPA, but DARPA say they say 1 to 8 can challenge to do so. Or you can say one is a driver utility vehicle at the site, travel this mounted across the rubber. Or fifth is a climb in the cell ladder and traverse industrial walkway. So maybe, maybe not. But anyway, that's a kind of thing. It says the DARPA challenge here, and that's you can challenge. Okay, so from the viewpoint of biomimetic, inspired robot system. Other like here, this came from a kind of DARPA challenge. They say, why don't say that like that? Close a valve like that. This is from DARPA. Okay, so they said. Also, U.S. is, I mean, did very nice. You know, right? National Robotic Initiative there. So that's a synopsis, very interesting. So that's co-robot. Co-robot here work together, something like that. That's a very interesting kind of funding here is. A small, large kind of a project here, and then here like that, internal co-robot. So that's a medical surgery here, or such an unmanned vehicle, even there's manufacturing here somewhere. Also like here, kind of those things here, and micro nano areas. So that's a kind of very interesting kind of things there. So also like kind of EU also have a kind of nice kind of things here. FP7 That's a kind of ICT type of things, such a kind of robot, human work together, such a cognitive type of thing. This is a kind of book I wrote in 1994, or something like that. Such a kind of several robot system here. This is a kind of real kind of color with the thing. So how the kind of robot work together, so like that. So like kind of thing. So this is something say the reason why I'm interested in is something like here, like kind of a human in a biological system. The cell is here, cell come up here, cluster, then make a tissue organ, and then animal here, animal become group and community, and make a society, like a society of human beings. This is all line, last 20 years, never changed, okay? Likewise, here, the robot is also here, can make a cell. It's a unit, basic unit here, make a kind of a module, or kind of a robot here, or a group, can say a robot system, say a kind of a robot society, robot being, something like that, okay? I try to make this one. So this is a kind of a story why I started those things here. So my goal was like to make this type of thing, but when I started those things, write a paper for the first paper, 1987, I did not have a technology to make such a small robot. That's a big one, very small. I didn't have such a technology, 1987. So that's a story. So the reason why is I try to make this type of robot. Inside hatch is so small, but go inside like that. So like that, grow up. Now disassemble it, come back here. This is the kind of thing. Real world, what they're doing, like kind of old storage tank, what they do there. They have it's about 50 meter diameter or 100 meter diameter, such a big one. So they try to take out the sludge out of the bottom of the tank here, storage tank. So then the people go inside and small part, disassemble it manually, and then come here, the guy come up here and teleoperate it. Okay? So it's so dirty, okay? And not nice. So that's why don't you make this one like more kind of a nice way. So also manufacturing-wise, why don't we make this kind of thing reconfigurable manufacturing system? So they depend on the kind of task, depend on the job. They can change such a configuration. So that's it. So something wrong, they change it like that. So this is a kind of something like a self-organizing manufacturing system, which was about 20 years ago. IMS International Manufacturing Project, that's kind of we worked on those problems. So the concept of kind of CBOT is the same line, and simply I didn't have this technology to make those things so small, the size of cell. But this was a picture about 25 years ago. So now there is a kind of a technology now. That's today. Then we may have that. Why so? Because kind of decomposition of a complex system into small unit, you can have many things, like a kind of functional, knowledge-wise, task decomposition. That's a very interesting kind of topic to work on that. And then that's like that. So maybe kind of one robot here, many group robot here, and that's a kind of many robot society. This is a kind of like a kind of monkey like that here, one monkey, many like ant or fish or other things. Then come here as a colony society of insect, society of human being here. Likewise, we try to make those things here. How the kind of individual, how the intelligence come up here, or those things come up group with a good communication, with less communication, how they make such a decentralized robot system, for example. That's a kind of interesting kind of topic here, and particularly such a kind of cell, come up a kind of organ, individual, society like that. So that's interesting. So individual, maybe we can have one robot, the main robot here, maybe we can go back to this here. That's a kind of thing. So anyway, this is a kind of all the things, self-organized robot system here, and this kind of picture doesn't change last 20 years. But still it was, okay? So this robot system, those days, still is a kind of thing, it is still kind of bad in a way, kind of engineering, physics, chemistry, maybe you can have a synergetics here, or fractal here, something like that, actual life here. Those are things, very, and we can talk about biology, those things. So this is a kind of thing, in 1985, made such a kind of prototype to show how they can make a docking, detaching, those things, how they can make those things, configuration, like that. So even made such a kind of manufacturing, flexible system here, so that kind of flexible transfer system. And then it was used in the kind of factories, as a matter of fact. So they all ran up here, changing the line, depends on the kind of task, depends on the kind of number of demands, they change the number of robots to work together here. So anyway, even like you like a Kiba, now it's a kind of bought by company, book, Amazon, okay? But this is in Boston anyway. So this is a kind of also kind of a multiple robot system, a simple way, that's a very work so well, like a soccer game, okay? So well, like that. So now, likewise, as I said, those things can maybe, now we have a technology. Maybe we can make it. That's why I started to make those things so here. So maybe we can have a kind of a assembly of a 3D cellular system. Those days, it's a kind of inorganic. Maybe here, organic devices. So we can make it some, or you can hybrid those things. So maybe we can do that. So this is a kind of thing here. So try to do this. So maybe here, something here, maybe we can make a kind of 3D construction somewhere in vitro environment. In vitro means like kind of in your lab, in my lab, in test tube, or something like that. So that's a kind of thing. So today I'm going to talk more like here, such a kind of 3D scaffold for vessel. This is just a mention about such a kind of simulator. This is also biomimetic, it's just micro technologies, and then hydrogel fiber. And just today I omitted here, hydrogel fiber. Then we can talk about kind of also assembly by magnetic hydrogel. Here I talk over here and here 2D or 3D cell assembly with a photo-linkable resin. There's a polymer over there. Also, like I say here, another like a single cell manipulation. A nano tool here. That's the thing. Maybe I talk about it. So like here, just briefly, just one picture. This is a kind of fragile coil, which I use for kind of DDS, drug delivery system. Didn't work, sorry. It's a kind of rotation. This is a fragile mode. Just on, off. I can control the speed of rotation. So like that. So if natrium is on, then they speed up, but like 7 or 8 hertz, something like that, rotation. And the other is like I say, kind of the bacteria here, so they rotate in this way. This way is rather not easy, so they rotate in here. So we can measure such a force, nanonewton level. So first of all, I got to kind of assembly method for 3D structure by photo cross-linkable resin here. So first of all, this is kind of, we can make such a kind of pattern here, and here then we can have a DEP, dielectrophoresis force here, so that we can make a kind of pattern. Then we make it this one. To make it fix those things, okay? So first we make such a donut like disc like that, and put it here. They can sell it here, and eventually make this kind of disk here, maybe, and then maybe in future, maybe people can make a muscle, or some other people using us making also muscle here, also like a kind of blood vessel here like that, also like a nerve fiber here, like those other people mentioning such a kind of neuron here. So maybe those can get together, so that's a kind of nice in the future. So the in vitro realization in vivo environment is something like that. So once again, the idea is always same as a cellular robot system here, like an individual single cell, group cell, and tissue cell level here, work all together. So make something like that. So make it come here, here, something come up here. So this is a kind of here, for example, here, then we make such some we can plant here with a 3D scaffold here. So maybe we can put it back here later on from laboratories. So this is a kind of in vitro realization back here. So that's a kind. We can make many things working here, those things. So once again, what a kind of strategy-wise is just to use such a kind of this kind of simulation of biological system here, plus analysis of assembly of a biological system here, such as a DEP, or kind of patterning here, fabrication, those things, also analysis of things, bio-bit analysis by nano tool here, make this type of thing. That's like the goal to make it. So to do so, we need some kind of small devices here like that. So we call nanosurgery, nano tool system here, nano tool here, the kind of diagnosis, extraction, cutting, injection, milling, something like that. So that you can try to have those kind of devices, nano knife, nano grip, whatever it is. So there are so many kind of 3D cellular system back so far. So like I can say it's a kind we have a functional tissue or in vitro environment. Maybe we can apply to such a kind of regenerative engineering, bionic simulator, or investigation of cellular system like that. Maybe so far the example-wise is not say like a kind of porous scaffold to make a your kind of blood vessel for tissue engineering here. Also like a molding here like that. So it's each of them is a cell, okay? It's a cell here in hydrogel, okay? Come up here. So also like a bioprinting, you know, like an inkjet print, like I say, just printed cell with a collagen. So that here. And also free dynamic, so that people can use kind of a hydrogel here. So that's just an example how you can make such a kind of 3D cellular fabrication assembly method. So maybe once again, you say maybe we can make it here, this pattern here, and the photo cross-linkable, and then we can make a microstructure here, then we can pattern by DEP, so that I try to make it once again here, okay? So, and then what we want to do is here to make such as like here, on-chip microfabrication, on-chip micro-assembly here. If you can do that, that's nice. Okay? So that's the thing. So like to do so, we need a kind of cell sorter, cell culture, cell analysis, cell can harvest here. So that it's all to go, such a thing. Okay? So this, we need a kind of precise assembly of 3D cell structures. So this is something here. So once again, project is here is a cell. Here we make a scan cell here. Then the electrophoresis is a pattern here, putting a cell here. Then we have a kind of pattern on chip fabrication here. They put it here on that. Then we assemble it in a way like that. If we can do that, so nice, but it's difficult because of this kind of structure. Okay, so if you just make a structure, we can make it probably layer by layer, like a cell seed. Okay, cell seed we can make it, but the question is that if it's thicker than 200 micrometer, 300 micrometers, then you need a kind of more oxygen. Because of lack of oxygen, there's a kind of cell will die. So that's why we need a kind of sort of kind of blood vessel here. Anyway, we can make such a kind of chip here with those things here. So this is a kind of pattern made by by SUT. So the pattern is, so this is a kind of yeast cell here. Then we see that how it look like. So this here, and then we can here. So that we can collect such a kind of a cell here on the electrode here. Switch on, switch off, switch on, switch off, like that. So, or even here, this flow. Here, this is no. Here, the flow direction here. But this part is no flow. So no flow, but switch on, boom. We can have such a kind of cell here on the line wise. So like here. So maybe we can. Okay. So we can make it here. So even we can make kind of such a we can make a microstructure to fix such a cell or here on the spot by EUV. Here, so as I said, we make a donut structure, microstructures on the spot, on the chip, okay? So that we can make it. It appears. So maybe you can see the donuts here. This is a disk here. So we get a pattern here. That's here. So also like here is an even flow is there. The flow is here. So maybe you can see this kind of cell here. Surface PDMS here. You see some donuts here. Okay, this is the kind of time 2 to 3 seconds, something like that. But anyway, we can make such a kind of microstructures. So that if we can have a kind of microstructure here on the top chip here, so that we collect such a cell here, and then we make such a disk of those things, like here, like that. So this is kind of culture cell inside structures. Then we make this one PDM as here, and then we just move in this way. So if I make like here, then we can make kind of a microstructure by PEG-DA here and put here like that. Like as a pattern, maybe barrier, so just a kind of 100 micrometer here. But anyway, this is a kind of structure here. So we are making those things going inside here, here like that. Keep going. So this is a keep going. So those are going on, so like that. So next one going on. So this is a kind of structures. Here. Then those can donuts go here, okay? Donuts here. So also there other way to make a kind of pattern here, such a thing. Oops. So a kind of fluidic chip here, go over there. They're flowing here. This is just a kind of electrode. So by electrode, switch on, switch off, then it goes through, but switch on and sorting in a way, this way, okay? So this is a kind of thing. So the stopping, go like that. That's a kind of things. So this is a pattern here, like that here, and like that. So this is a pattern and controls those things here. So maybe we can see. the kind of cell moving here, microchannel here, so the whole thing going on here, go here, then switch on here, then go here this way. So we can control so those kind of beads here, so like here. So the idea is of going here and here. Also we can make a kind of laminar flow here, this way we can make a line up all the kind of cell. So that if once we get on so line up with a gel wherever it is, we can fix it, we can make it some microstructures. Oops. Sorry, the video is broken, link is broken. But anyway, try to make those kind of things what I explained here. So maybe this works. It works. Jeez, this is a video link was broken here. But anyway, we try to make kind of microstructures, the beads here like that. So that's a kind of idea. Okay. Anyway, that's a kind of, once we make a disc like that with beads kind of cells here, we can make a structure like that. So that's a kind. So also like this is a kind of this is just a kind of with a manipulator. We can pick up such a kind of a ring here. This is a ring here. How we can put the ring? This is a ring, okay? Which just put in a small tab here. I pick up something here. So this is manipulator. So put it together. So ring, so small ring, okay? This is just a kind of demonstration. This is fun. We don't do that, this one, but because just showing us to show such a capability of manipulation. This we don't, as I said, we don't use this one, okay? So main manipulator, sub manipulator. So pick it here one by one. So like that. Oh. Okay, this is 8 times faster, so don't be like that. Okay, so just pick up one, and like that. So this disc of the cell put together. Eventually, we can put many links here. So many, huh? Okay. Then like you can make a kind of like here, like a small micro shish kebab, something like that, so that you can put here, right? So this is a ring here, but this is a cell here. So try to do that. It's alcohol and inside. Just again showing such a capability. So anyway, to do so, what do we need is a kind of characterization of cell. What's that? What kind of thing? So maybe we need a kind of micro nano measurement. What's a cell work together, like a dynamic interaction between multiple cells, cell and cell together. Nobody knows how the kind of force in between, such as adhesive force, or some kind of micro nano manipulation, like injection, suction, like that. Micro nano assembly, like a construction of artificial cell, like that. So that's a kind of thing. To do so, we need something. What is that? So need something like this. So maybe we need a kind of micro nano here. That's a kind of micro nano robotics get dry, wet areas, millimeter wise, micrometer wise, nanometer wise. So you can make as many kind of things. Say like here, this is wet conditions, nanometer wise. So you can this is a cell. You can measure such a kind of stiffness, or kind of Young modulus like that, something like that here. So this is a kind of interesting areas. One example, like kind of single cell stiffness measurement. What happened? Like say, like a kind of, I showed such a kind of blood vessel simulator. The blood vessel simulator, that's a kind of a young modulus is a 1 to 3 megapascal for human being. This cell is also same 1 to 3 megapascal, something like that. This is very interesting finding here. So anyway, the thing is we're going to organic, from inorganic to organic level here to make such as here. So micro nano assembly technology, that's it. So that I integrate such a basic level, micro nano area, to actuation, assembly, to make more like that. If we can do that, very fine. But we are still on the way here, but that's interesting, and we are working on that. So micro nano material, look at that. Here, there are so many things here, like kind of thing, okay? So all kind of same areas, sensing technology-wise, there are so many others, like technologies, okay? Also like a kind of actuation technology-wise, there's kind of optical driven, or magnetic driven, there's PZD, all the things. Those are things here. There are many work going on those areas. Also bioassembly-wise, there are so many things here, okay? Then that I showed the kind of EVE project, such as EVE is a kind of a human body simulator, which I made here last 20 years. And finally, we made a kind of simulator and put in the market 7 years ago. So this is similar, as I showed over there. What we can make is just take a kind of CT like that, okay? Then CT data was sent to laboratory. Then make a kind of vascular model here. Then it can inkjet-wise pattern, pattern. It's like a silicon. They make this type of thing inside. That's a kind of thing. So this is some interesting things here. Those things. So this is just I showed you before. This is kind of this is silicon and transparent. So that's easy for medical student to understand. Second, the kind of medical doctor can have a kind of rehearsal before the surgery, because this is a tailor-made kind of surgery, okay? Everyone have a different thing. And also like kind of the medical doctor can pass exam to get a qualification to this kind of surgery. That's a kind of thing. So I wrote a book about such a microsurgery, those things also like here. So this is a kind of interesting application of microtechnology to the medical areas. So anyway, this is a kind of thing. Since we have kind of say EVE here, such a simulator, whole body simulator, eventually we want to make a kind of super simulator here, which super simulator is a kind of biomimetic those things, bio everything, such a like say like kind of immune simulator also. If you can put here using such a kind of micro nano or biosensors like that, that's fine. That's a thing. In between, it took a time. People cannot wait. So that's why we here perhaps we can make. We already made this type of scaffold for tissue engineering using such a CT scan. The same technology of making this kind of things. Then try to make a kind of blood vessel. This is a kind of first step. Then go to the organs here. Then eventually make those things. You know, this kind of human body, such a long blood vessel, or say it's almost 30 kilometers, almost length of circumference of Earth, the Earth. Okay. So this is a thing. Eventually, to make a super simulator. But before going to be a super simulator, we need some kind of biological cell tissue and what. So we need those things, okay? So also need a kind of micro nano kind of technology here. So that we once again we need this one here, implant those things. So this is so we need a kind of such a technology kind of say like a SEM, like a ESEM, environment SEM nanomanipulation system, so that cell can survive, alive, okay? And this kind of, we can make a nanotool here from dry areas. So integrate over there, put it here, such a kind of single cell, as I mentioned those things. So like in diagnosis here, come here. So all the kind of a nanotool, nanosurgery come up here, bring back those things here. So this is thing. So single cell, that's a kind of thing. Nanosurgery for the characterization of the cell. That's here, maybe injection, injector, extraction, or the kind of nanopipette, nanofork, nanoknife, those things. So that's a kind of thing here. So maybe we are talking about this area, okay? Eventually come up with this cell here, this level, like that. So this is the kind of things here. In vitro realization to do in this way to go. So that's kind of nanometer scale, micro scale, millimeter scale, scaling to do so like that. This is just example of such a kind of making a platelet in your blood, so like that. So anyway, this is a picture here. So maybe we have to go here where we are somewhere here like this, then go over there. Okay, so like that. So this is just mentioned. So this is here. So now I just show kind of ECM manipulation inside the ECM here, so that here once again that to measure such a kind of such an adhesive force here as a cell sticking on. So made us this type of kind of fork here, and put a kind of 100 nanometer pattern here. We put a cell on that 100 nanometer pattern on the substrate. So this is a substrate. This is a cell here. See that kind of magnified in this way, and they made this kind of manipulator here. So thing. So measure. So this is a kind of fork. Aligned here. The nanofork come up here and pick up those things. So pick up everything. So if you pick up, then if it's a kind of cell will resist against such a kind of fork to lift it up. So that's here. So like that, sliding, lifting up, lifting. So from the deformation here, we can measure such a kind of this force, maybe other force. In this case, about 500 nanonewton like that. Okay, so maybe like that. So also like kind of a cell-to-cell adhesion force here, that's a kind of also interesting to measure those things here, those things here. So put here like that. So here cell picker here, pick it up. That's we measure the cell. So if the cell and cell get together, maybe we can. So we made such a kind of microprobe here by using FIB etching here. So that's like that here. So this is a kind of picker here. So this is a picker here. So but here, something like that. So try to move over there. So the last moment it lift up the gap here. So then we can measure such a kind of force here. This is about this is about the kind of 50 nanometer, something like that. But anyway, time-wise, here this is a time, a minute here. So once put it here, then the cell and cell get together. Then the adhesive force increase like that because of the kind of protein like that. Okay, tangled. So at the beginning it's okay. This is a kind of saturated in a way, about 25 nanonewton, something like that. So this is a cell by cell. In this case, 25 nanonewton here. So this is a kind of explanation here, beginning here this way, then later here and here in this way. So that's a kind of explanation here. And also here, how to cut a cell here. Of course, you can cut anyway, but maybe we can cut something like that. Maybe you can use diamond knife, but also you can use a kind of CNT carbon nanotube type of. But it's so weak. So we made this kind of single cell cut knife here like this. So like that. So put a kind of cell here. Try to measure it. Once again, the force kind of from cantilever-wise. So that from the deformation, we can measure the kind of a force here, how it rises. So this is a picture here. So come here. So bring it here, like that here. So here. So it deformed here, so that we can measure those kind of stiffness. Also force too. So one cut here, and also maybe this here. So this of maybe a certain degree or something like that, depending on kind of sharpness of those things. So from deformation, we can measure those things here before cutting, after cutting, like that. We deform such a. So in this case, we use a kind of vibration. With the vibration, then we can reduce such a force almost half, like that. So oscillation like that. Okay. So that's a kind of thing. So time's up. So anyway, that's the thing that as I say, the living machine. Maybe we can make it can bio-inspired, or can biomimetic approach like that. So maybe we can make it, or in robotic side, or even like a kind of such a kind of hybrid biosystem too. So also cell assembly to biohybrid device and system, and single cell analysis and characterization based on ESAM nanomanipulation system, and single cell nanosurgery system include many kind of devices, nano to handle those things. So that's going to conclude my talks, and I think there's a lot of futures to make those things possible. Thank you very much.
All right, fantastic. That's the future of nano and micro robotics. So questions.
Living machine.
And living machines, yes. You have questions. Well, let me start with a question that always I cannot really get my head around. Like in some sense, what you're showing us is how slowly components of such a micro nano machine will become available. Yeah. But certainly, if you operate at that level, they will need to have a lot of autonomy.
Yes.
So how are we really going to deal with the issue of control, sensing, and actuation?
Yes. So this is a kind of assembly. So then we need a kind of in the picture I show is a kind of nervous system for so kind of intelligence, like Ken Jones mentioned yesterday. Other people like also like kind of those things. First of all, we need a kind of survival is such a structures. To do so, we need a kind of delivery of nutrition. What a nutrition delivery? That's of course, but in culture, but not way better. We have to use such kind of blood vessel, capillary. So that's a kind of have to have such a thing. First of all, we have to kind of make such those things, and then it will grow in a way of self-organization. That's the kind of you're expecting. But the thing is, so far we can make is about, as I mentioned, about 200 micrometer to 300 micrometer thickness. That because beyond that, you need such a kind of blood vessel to give nutrition.
But then how do you see, what do you see as the first, let's say, breakthrough application with technology? Which specific application?
Specific application, first of all, best way is to make such a blood vessel fast. Even today, blood vessel, artificial blood vessel, less than 6 millimeter diameter is not so easy to real application. Okay, you can find kind of artificial blood cell with a size of 6 millimeter diameter everywhere. You can use it in the hospital today in practice. But the smaller one, it cannot make it. So if we can make those things, first of all, a little short one here, even here, okay, right here, then it helps a lot. Okay. Yes, that's a kind of first kind of. So that's it's very interesting to work together with medical doctors, also like biologists. That's a kind of combination of the three disciplines: biology, medicine, and engineering together.
Other questions? Okay, so if you have no other questions, then thank you very much again for your presentation.
Okay, thank you. Thank you very much.
And then I have, so right now in our program we have the road mapping event, and this is actually very relevant for us, and I hope also for you. So we know now that the coordination action that's supporting the conference will, with very high probability, be continued. So this also means that the European Commission is supporting this whole idea of building a community in Europe of researchers in the domain of biomimetics and biohybrid systems. And one of our tasks is, and this not only to build the community through events like this, like the Living Machine Conference or the BCBT Summer School in September, to which you also are very much invited to join, but it's also to get an understanding of what this field is about, what is the future of this field, so that also the European Commission, in particular its Future and Emerging Technology Program, can try to define calls to support research in this area. And in some sense, the European Commission has been very open in defining these kinds of challenges, because they basically say, look, it's up to you guys to tell us what your field is about. It's up to you to tell us what we should be funding and when. So it's for that reason that the coordination action is also very much focusing on these road mapping events. And for that, of course, we want to get your input. And so Nathan will guide us through that as well. We will do that together with our experts. So Nathan will tell us who will invite us here on the podium. But before we do that, Anna, you want to say something about the dinner? Okay. And then we have our dinner this evening at the beach in the restaurant Boo, and you will get directions to that after our road mapping session. Exactly, you got it. Nathan, you want to have this stand in the microphone? Okay.
I don't mind.
Mm-hmm. Well, first, is the cable in there already? Okay, then you have to switch. Did you switch it?
I didn't set it.
Oh, there you go.
Oh, okay.
So, you want to give your example now?
I'll give the example before people give them so that they're directed. Yeah.
Okay, so the idea is, actually Nathan, who is working with Tony, has been spending quite some time on developing these roadmaps. And so to get the discussion going, he will give us a first example of how you could think of such a roadmap in a very specific area of haptics. This is not necessarily to say, look, well, haptics or whatever else Nathan will say is our target. It's just to give you an example of the kinds of issues and problems we're after to sort of guide the discussion. So after that, it's really up to you then to start to generate some keywords and some ideas that make roadmaps in this broader area of biomimetics and biohybrid systems.
Could we do the same as yesterday and have people sat up on the stage who are the plenaries, if that's okay?
Sure.
Are you guys okay with
So
coming up
plenary
here?
speakers up here. Mm-hmm.
And also Tony? Tony, would you like to sit up
Tony.
here as well, please? And Frank? Please?
There'll be chair dance soon, no? We need more chairs. Go.
And anyone else who feels they should be up here?
We're going to get a few more chairs, don't worry. But of course you make it difficult for them to see you what you're going to talk about.
Well, it's okay. Maybe I'll start.
Yeah.
So, to try and enthuse you why that biomimetics, the subject that we all work in, is going somewhere, here's a plot of publications in biometics over the last 15 years or so. As you can see, the area has literally exploded. Now, usually when I show this plot, people go, yeah, but isn't science expanding as well? Well, so I've looked up the amount that science is expanding in general. Turns out that cumulative publications in science are doubling every 15 years. Whereas what we're seeing here is individual publications in biometics per year, not even the cumulative total, but per year, are doubling every 2 to 3 years. And have gone from an area which was relatively small in the 1990s until an area which has well over 1,000 papers a year and is continuing to grow. Okay, so, I mean, both Tony and Paul have explained to you the importance of biometric roadmaps, so I'll repeat what they said again. And essentially, a roadmap is a plan that identifies goals in the research area and is fed back to the policymakers so that they can allocate funding and trend-setting agenda. This roadmap in particular would be fed back to Cordis as an output of the Convergent Science Network that funds this conference. So it is in our interests to make this a persuasive document, because the funding, if it's a persuasive document and can bring funding into the field, that will be at the expense of fields outside biometics. We're not competing with ourselves here, we're competing with other areas outside. So it's in all of our interests for this to be a persuasive document, but it's also in your interests for you to promote your research area, because we're trying to cover whatever people tell us and put it, but if you don't tell us what your interests and your priorities are, then obviously we can't put them in the roadmap. So in particular, if you have ideas relating to the discussion today, just email me with the ideas, just a paragraph or a couple of lines, just with any useful information, just send it to me and I'll try and put it into the roadmap. Okay, so to try and sort of focus the discussion today on what we'd like out of you to put into the roadmap, I'll give you a little example of what I'm interested in. I'm a relatively new researcher to this area, so maybe these are my interests anyway. So the area I work in is tactile perception and manipulation. I'd like you all to tell me the area that you work in first, so that we can categorise your answers. Some example grants. I'm very interested in hearing about example grants in biomimetics. Here are a few example grants that in tactile perception. So research goals and priorities. This is what we really want to hear from you, because this is setting out what the priorities are in the research areas, in the specific research areas that you tell us, that we will feed back to Cordis. So 3 example goals that I consider to be important in tactile perception is integrating tactile perception with grasping and manipulation in robotics. Concrete applications of tactile perception and grasping in advanced manufacturing and assembly. And thirdly, human-robot interactions via touch. Humans are tactile animals largely. That modality for communication with robots is an important one, which hasn't been fully utilised yet. We're also interested in risks and ethical issues that you feel about for the research area. So, for example, in tactile perception, an example would be threat to the human workforce. So, for example, if we can build robots that make things for us, we don't need people to do that. So, what do those people do instead? That's an ethical issue. Okay, so now I'd like to move over to you guys.
Okay, so I got the microphone. So what I suggest we do, just to keep it simple, I will start here with Joe here, and we can move past the microphone. So I think it would be great when you guys can just declare, if you want, what these priorities are in your mind. Let's say today, where's the field now? Where are we today? Let's not go further than 3 priorities, but then also maybe declare where you think we should be going in the 5-year and that 5-year time frame. All right?
Keep it short, right?
Yeah, of course.
Keep it short. When I originally got involved in robotics, the goal of the robots was to clear underwater mines. Generally, the best use of robots is things that are either too dangerous or too boring for humans to do. And this is a huge problem. There's enormous amounts of unexploded ordnance that are left around as just debris of wars. They're a big environmental problem. The biggest problem in dealing with this is where the mines are in places that are hard to deal with, hard to predict, underwater mine or rivers, coastal areas, stuff like that. So I think the really important thing that we need to go at is trying to find ways to make these vehicles autonomous. And autonomy is a big issue. Just defining autonomy is kind of an issue. But I would point out that a common pissant is a fully autonomous system that operates with impunity in unstructured environments without training or supervision. We should at least be able to focus on something like that.
What area of research would you categorise those two? I mean, it's general robotics, but if you were to be more specific about categorising
humanitarian demining.
So my primary area of interest is in completely soft material robots, so developing the technology to make machines that are very highly deformable. I think that technology will also get incorporated into more traditional robots as well. Just to follow this list a little bit, the main funding that has been around for this recently, in the United States, the DARPA ChemBot program was a very, very big influence on developing the technologies. And here in Europe, I think the Octopus project, for example, are really good models, if you will, for the type of thing that needs to be funded. Well, lost the other bit. But when it comes to what are the major issues, I think the biggest issue of all is control. How do you design a system that can control massive, if not infinite, degree of freedom structures? I would argue that we clearly can do it because there are animals that do it. So there is a solution to the problem, and that is the direction I want to go in. The other major problems are developing actuators that work under those circumstances. So I think that's still, despite all of the development of EAPs and SMAs and all those other technologies, it's still lacking. It's really not where it needs to be. So those are technologies that are very, very important, the control and the actuators. I think also the ability to fabricate soft structures accurately. That's not actually easy to do. And the last topic, I think, was ethical issues and what we need to deal with. For completely soft material robots, I don't see many issues that are going to be abrasive for society. I think it's going to be a net gain to society. For the work that I'm doing with using tissues to make robots, there are clearly ethical issues with that. I think we can circumnavigate or avoid, those issues by not putting a nervous system in it, using computer chips, and by making sure that we use cells derived from species that we have traditionally not worried about exploiting, predominantly insects and invertebrates. So that avoids a lot of the ethical issues. But we can talk about that tomorrow too.
I'll be quick because Nathan's already said some of my interests, which are obviously related to the haptics and the tactile sensing work we do. I think a broader issue is the materials that people have mentioned. I think we have to think about how we can use materials more intelligently and build a bridge across to material science, biomechanics, and get them to bring their ideas to our meetings. Well, a lot of my work is in computational neuroscience, and I think what's missing in our machines is a capacity to not quite self-awareness, but to know more of what they are and to be able to remember more of their history and to allow that to influence their decisions. So self-sensing, awareness of your body, awareness of the space around you, all these things our robots are really lacking in, and I think that's one of the things that makes them stupid. And knowing what you were doing 5 minutes ago and how that should affect what you should do now is also something that makes them very stupid. And I think we can learn from natural systems how to do this effectively without necessarily going to really detailed neural models, but we can discuss that.
Okay. Myself, I don't mention about such a micro nano, but it's more like a robotics right here. In this case, perhaps kind of a robot for aging society. Mainly because somewhat like back home, also somewhere, some other countries, kind of age quake there. So going to be in next 10 years or 20 years. So we already have an age quake in back home. So that's a kind of good thing to think about. Then robot technology, maybe such a kind of human-friendly robotic system, like those things. Usually, we go to the robot, but in this case, robot have to come to human. That's a kind of concept. So, like, your car has to come in front of you. Okay, you don't have to go there, but the car has to come here. Because at home, the toilet must come to me. Not you don't have to go to the toilet. Toilet has to come here. So change such a kind of mind. Okay, that's a kind of important to think about. So there's such a kind of robot already in prototype. Toilet come. Maybe people laugh at me when I mention the toilet come. Mobile robot toilet. But simply think about it, like those people who take 1 meter per minute to move. Like with those people need such a kind of thing. Robot is faster to come than the aged people go to toilet. Okay, so that's a kind of, for example, everything must human-centered. Robot have to come. That's the kind of thing. So challenge, thing. This kind of ethical issues, yeah, there's a human here inside. So they are kind of involved. Those things. There are such a kind of ethical problem, but this is very, very human-centered, friendly robot system.
Yeah, I'm a little bit coming from the outside, but I mean, you saw from the talk just before that the whole manufacturing problem, if you want to go smaller scales, how you do that in a very efficient way. I think there's what we can still learn a lot from development of how creatures, how cells are organizing themselves. And I'd like to focus a little bit, how much you can, the bottom up on DNA origami, things of synthesis, how much you can come from there and try to connect to bottom up methods. There's a lot to be explored. I also think there's a lot of potential towards the medical applications. But at this point, I would not pinpoint directly to special things. A lot of technologies and ideas still, self-organization is not yet fully explored, and so let's see. I mean, I would, last time we sat together, it was very development-driven, and I would cautious a little bit and allow ourselves also some place to just play around and just be curiosity-driven. But that's probably not what the EU wants to hear from us. I'm not sure.
Well, we can tell them.
So my interests, I was trained as a computational neuroscientist, and I started building biomimetic robots to build bodies to go around the brains that I had designed, the computational systems that I designed. And I was always interested in the impact of the environment, the actual physical environment. I had an ethological training, so I was interested in those impacts propagating through. And my interests haven't really changed. I think of biomimetic robots as an important tool for being able to understand the ways in which nervous systems organize and coordinate behavior. And I think that's the central goal in my interest in the things that I do, is to use them as ways of testing hypotheses about how brains work and validating them. And I think that should be a core interest in our activity as a community. A lot of what we've talked about yesterday and today has focused on developing the technologies, but I'm going to again emphasize the idea that there's this cycle in terms of the biomimetics where we build the robots, which may introduce new technologies, but they also tell us more about the biology and the organization, control and coordination of the brain. And as we cycle back and forth between those two, we narrow in on the truth about how bodies and brains and so forth organize behavior. I think that should be a goal. And one of the priorities that I think we should have is in recognizing that the animals that we study, the organisms, the systems that we study live in the real world, And we should put an emphasis on trying to build robots and systems which can actually be competent to work in the real world as well, rather than just in the laboratory, turning something loose to be able to meet the actual challenges that the animal has experienced.
Yeah, hi. So I thought it was interesting that you picked manipulation and tactile sensing as kind of a focus area. It's interesting to me that for more than 30 years, it seems that tactile sensing has been lagging behind vision for many good practical reasons. And when you look at even very simple animals like a slug or a worm, the number of mechanoreceptors they have per unit area is so much greater than what we're able to do today in robotics. And in the end, it largely comes down to what we can fabricate. The things that we're building today with silicone rubber and urethanes and a handful of embedded sensors are not even close. And as we learn more about how biological systems respond to tactile stimuli, the need for better manufacturing capabilities becomes all the more apparent.
So I've heard many great examples of where we should go with our roadmap, and I would like to add something at a different level, and it's more educational. I think a major challenge for our field is the integration of biology, physics, and engineering. If you talk with an engineer or a physicist, they typically don't know much about biology. If you talk with a biologist, they typically don't understand basic textbooks that are really trivial for engineers or physicists. And this is a real problem. So the communication between the different communities and also integration. Just think of a review, someone making a wonderful contribution to our area, but fails in one of the sub-areas to some degree and doesn't get the respect for still significant work. I think this is just like both from the scientific point of view, but also educational, like how do we help our students to cope with these challenges? They effectively have to be 3 times as good as one student has to be in one of those fields. So that's a challenge.
Yeah, I definitely concur with many of the points here. My approach is particularly in the area of trying to take highly specialized nervous systems in the hope to make them more tractable. So in terms of example grants, we did one of the first grants with, say, taking ideas from the moth in the framework program about 8 years ago now to translate that over to chemical search, and then you saw another example today with another specialized system in terms of maybe giving us solutions to chemical communication. So my belief is that it's important that it represented these very highly specialized nervous systems because of the key point of constraints, sufficient constraints in the system and very compact and efficient solutions which may give us low power and effective and highly robust engineering answers. And a good example of why we need these types of things is precisely today in my talk where so I plug in my projector and of course nothing happens and it freezes. But as a human, yeah, well, it shouldn't be down to me to intervene in that. I mean, both this projector should understand what a computer is, and it should also know, the computer should also know that the projector is in the vicinity, and it should also know, it should have a degree of intentionality about its behavior in terms of what it needs to be doing in terms of its existence in the world. And when you look at all of the technologies around us, not just this particular example, but this is precisely what fails in technology, is that it's totally dependent on humans to fill in the point of intentionality between in terms of this sort of missing purpose. It should already have a purpose. It should know what that is, and it should know how to fulfill it. And these examples of specialized nervous systems already have very precise answers to these in terms of intentionality. So I would push this as an important area where maybe these specialized nervous systems can give answers.
All right, we have quite a portfolio, but now also to motivate the rest of you, of course, was all wrong, right? So feel free to add and react. And indeed, if I could add something else, because sometimes we have, let's say, the idea of targeted systems. We have the idea of component development or of, let's say, long-term application development, like a robot assisting the elderly. But I think an important challenge we face is always this issue of the system integration, that it's not only about the components or building the robot or studying a piece of a brain. I think a real challenge is to look at this from this systemic perspective where environment, body and brain really comes together. And I think what we really have to think about is also more a psychological engineering of these kinds of systems. I think we absolutely have no idea about how to really do that. So, and sometimes also in robotics, you feel that the robotics becomes a bit over-engineered because actually no one has a clue about how to really do the control in a smart way, how to give these systems memory, like Tony's saying, and so on. And I feel that to think also a bit more about the biomimetic form of psychological engineering is really lacking right now. It's also a bit what Tim was sketching, right? That's really one of our frontiers. So here we have a portfolio of issues on the table. Anyone in the audience wants to react to this right now? It's your future, right? Yes, please.
Asking not to concentrate the control issues only on the nervous system, because there are some organs that they can make the decisions and coordinate the behavior without nervous systems, like plants, for example, and also bacteria, I don't know. And there is coordination, there is actuation, and there is some kind of control. So, but it's not only nervous system, just this. Thank you.
All right. Anyone wants to react to this? No, maybe there's already a first reaction to the plants and nervous systems. Anyone wants to say something?
Yeah, just to reply to that, I absolutely agree, although I'm a neurobiologist, and so that's clearly one of my focuses. You know, I am of the firm belief that you have as much computation built into materials and structures as you do in the nervous system, and in some cases it may be the dominant form. The two work together, and a lot of systems can probably be extraordinarily smart. I don't like to use that term, but very, very smart just because of their complexity, their ability to adapt to all of the conditions they're likely to encounter. You see that all the time in evolution, so I think it's a good point.
But now it is the case, there's some discussion about plants having something like a nervous system, right? So it's not like a binary decision, but that's a good point. Holger. I think you turned off the microphone.
Okay. It's supposed to be on. Is it on now? Okay, good. So I think your point with systems integration in combination with control is one of the major issues for more complex robotic systems which are self-aware and so forth. And I think this direction is very interesting. But don't we get ourselves into some sort of dilemma? We want to have completely autonomous robots who can actually anticipate what the human wants, and I agree actually on Tim's points he was making. But how much of autonomy do we really want, and when does it actually become an ethical issue? So is there... Oh, there we go. This is a real microphone, no? So then the question is, what sort of safety net are we going to plug into that?
So it's really an issue, an ethical question.
It's an ethical question because, as much as most of you, I would love to push what you can do in terms of autonomy with robots, and I would like to take advantage of the fact that forward models and whatever e-print copies are massively useful in creating something like awareness or an internal representation, which is very helpful in terms of control altogether and fostering autonomy. But where are the limits, and what are our measures to keep it at a certain line?
Right.
Can you say what you think the line is?
Oh, well, that is probably an ethical line that everyone else, every individual has to decide upon. But I mean, I think it's more an absolute threshold, I would say, which is there must be always a way of, shutting it off altogether. There must be the emergency switch off.
Tony is asking you to indicate to us how far from the threshold we are now.
Well, no, my question was...
Oh, okay.
So I just wanted you to clarify what you think the risk is, because ethics is about identifying risks, and so the risk is machines that go out of control. Is that what you're saying?
Yes, basically that is. Well, I mean, I guess the most powerful tools we try to implement is, say, self-learning networks, control that can without any supervision, really improve the performance of the systems. But at which level, and there is a level there always, where suddenly the machines are doing things we do not anticipate them to do. And I cannot give you the answer where I would say, okay, oh, this is a little bit too much. Because at the same time, I'm fascinated, of course, of the potential there is. So it's a tricky one. It's a little bit of a...
So if you remember, the basic control architecture of my robots were based on what we call search vectors. And the search vectors might be to go out and search for a mine. They might be to go out and pollinate a flower, for example. Now, the whole idea of this is what I call supervised reactive autonomy. And it's analogous to, say, if you took your dog for a walk, you're the supervisor. If you throw a stick out in the lake and the dog swims out and brings the stick back, it's reactively, autonomously doing that mission, but it's under your supervision. Now, if, for example, while it's going out in the lake, it finds a mine and reports back to you that there's a mine, you might give it a message to go out and park on the mine with a detonating charge, for example. So the whole idea of this supervised reactive autonomy is that there is always a human in a loop exerting some sort of supervision over the process and making the decision that we certainly wouldn't want a machine to make. Frank.
I think you've struck on something that's terribly, terribly important. If we think about our biomimetic program being successful in imitating animals, we know there are animals that can harm human beings. There are plenty of them out there. And if we give these agents autonomy, we have legal questions about who is responsible when a human is injured. I think that's the thing we need to have a perspective on. Is it the person who switches on the device? Is it the person who designs the device? Or is it, as the case is with an animal, is it the animal who's responsible who has some kind of intermediate legal status within a human society? And I think you're pointing a finger down a long road. I don't think we're anywhere near needing to worry about that. But if we want to explore these possibilities of making our machines have the properties of animals, we will face it at some point.
I just want to come back to the, if I'm allowed to switch gears shortly, to the interruption initially we had. We saw a lot of experiments with rats running around, and I had this movie of these white blood cells searching the bacteria. I think the potential to select out cells for specific targets, for specific things they're doing, is just starting to develop. So people are just starting now to set up experiments to do large-scale screening for, micrometer-sized cellular robots which do what you want. And because the bottom-up approach there is very difficult, but that could be also a potential thing to target.
Now Tony has become an expert ethicist in the last year, so I'm sure he has an opinion on this.
I don't know about expert. I've been leading a working group on ethics for the Robot Companions for Citizens flagship pilot. So I've been doing a lot of thought, and I've been talking to people in ethics. My view is that there are a lot of ethical issues that they aren't ahead of us. They are here now. And for our field to grow and be successful, we need to think about them continuously. We can't put them off. I mean, I think you just have to look at what happened with GM food to see what happens with the community that think they're doing science, and in fact they are actually doing something which potentially transforms society. And when the news gets out, people reacted to it very badly, to the point that in the UK hardly anyone will eat GM food, even though they're perfectly safe and very good for us. But the science was presented terribly badly. And people are concerned about machines, and especially living machines, it's going to be a threat. And I think we're our own worst enemies, and that some members of our community will go out and say that the robots are going to take over the Earth. And we have to really think about that threat and actually evaluate it. And I think that the threat is relatively small, but we do need to think about the risks, and there are many others that we should think about, not just that one.
Another, and there's, of course, we can spend a long time talking about ethics, right? But there's another side to this, because you can also say, look, can we actually allow these machines not to be autonomous? Can we afford them to not have autonomy within certain contexts, right? So sometimes, and that in itself also then raises another ethical issue, because do you really want to deploy stupid machines in your environment, right? Is that really what you want to do? So it's not just by giving them autonomy you create the ethical problem, but also sort of preventing them from being autonomous, you might create other kinds of ethical issues. So I think whatever we do, we face this ethical challenge. And what I take from that, also from the reactions, is maybe when we talk about our research goals, we should maybe already identify also their intrinsic ethical implications and make them part of the research agenda, as opposed to some add-on, right? Like, oh, and now we bring in the token philosopher who will say a few words about ethics, right? So it's really also an intrinsic property of what we do. We have to be aware of that. I completely agree. And then when you bring up the word sentience, for instance, or consciousness, then feeling comes in, and people are very worried about feeling robots and suffering robots and so on. So the ethical agenda is a big one, okay? So are there other...
Yeah, it seems to me that, well, if you compare smart versus non-smart, or I think a good example is whenever you talk about a bomb. I don't know why that makes such a big impact for people, but it definitely does. So if you think about smart bombs, for example, people worry a lot about that. Whereas if you drop a bomb, they worry about someone dropping a bomb. But as soon as a device like that becomes smart, it's dangerous to us. So I definitely think to actually provide, if you put on some sort of smartness to your device, that is a danger for society. People will start worrying, but I also worry about the discussion being ahead of time as well, because otherwise, could really stop science. I think that is a discussion that we should kind of have, because is it important to beforehand warn people that we're all talking about, you had a great example, you wanted the beamer to think about what its purpose was, right? It was a great example that we're all thinking about these kind of things right now. Should we warn people about that, or should we first build it and see what the consequences are?
This is a good point, right? Because before you know it, you end up in this whole singularity hype, right? Where you say oh, the robots will become smarter than us, and they will take over the world, and I'm warning you. So now give me all your money so I can have a research center that will protect us from the robots taking over the world, right? So indeed, we should keep the ethical considerations in balance with the science that's actually going on.
Yeah, so I mean, we shouldn't talk about ethics all night. I mean, I could easily. I think it's about how you do science, because when you talk to ethicists, they don't say, I have an answer that what you're doing is right or wrong. What they tend to say is, have you done any kind of risk analysis? What are the benefits compared to the risks? Are you aware that the likely negative impacts are things that we probably can't predict yet? So even when you've done your risk analysis, there are still worries. And it's about responsibility as scientists, ultimately. Are we responsible about the way we present our science honestly in terms of its limitations and potential? I think we tend to over-exaggerate the potential and not tell people about the limitations, and that's something that can stoke anxieties. And I think we should also be more open about the way we do our science so that we can show people and we can see exactly what we're doing. And we need to counter the singularity hype because it's potentially very damaging to our community. We could end up closing down the whole area of research because people are so worried about this.
Yeah. I want, if some of our topic of interest are concerned with self-awareness, as Tony has mentioned, behavioral regulation or learning, so I'm wondering, do you think that cognition has its place within this roadmap? So that's my first question. And just concerning ethical issues, I think it's never too soon to raise them. And historically, it has always been the contrary, that we realized afterwards what are some of the problems related. So I think it's never too soon to put the things on the table clearly and raise the right questions. And
so cognition, I've got this one. Yeah. So cognition does have a place in this roadmap if it's got a concrete goal associated with it in terms of research. So if there's a robot that needs to do cognition for some specific goal, that's how it would be best placed in the roadmap. But I think an abstract argument about cognition would not necessarily have the same weight. So it'd be good to have some sort of specific goals that you see as being important for your own research and biometics in general. Which has just stunned everybody to silence.
Just to emphasize this, I mean, cognition is unavoidable, right? So if we talk about also, Tim wants to have the beamer to actually understand him. Now, this might be beyond the kind of cognition that we know, but still, to understand Tim, even that would in the end be a form of cognition, so it's unavoidable, right? Absolutely.
Yeah, I fully agree with Frank, and I think that it's important to have room for fundamental research on biology, and because it's a cycle in bioinspiration, for example, in biorobotics, it's very important to go to biology from robotics and from application, from robotic demonstrator. So I think it's important to have projects that have not always application or technological application. It's important to have fundamental research in biology because, for example, in France, it's getting harder and harder to fund fundamental research on, for example, on insects, on behavior in insects. And I have in mind this very wonderful study on how mosquito can avoid drop of water during flight. I don't know if you know this great study. And I think it's almost impossible to fund such research in France now.
So it's on basic research.
I think the thing we have to watch out for are kind of the unintended consequences. Somebody, I think it was Tony, mentioned genetically modified food. And the problem there is not what it does to us when we eat it. At least in the US, the problem is that it leads some farmers to dump massive amounts of herbicides on their fields because they know that the genetically modified foods are resistant. Meanwhile, there are all these herbicides. So where are we with things that are more autonomous? So we're just at the point now that in the state of Nevada, we have allowed self-driving cars. We have no idea what the unintended consequences there are going to be. Actually, I can make a prediction. The unintended problem is going to be when humans decide that that any time they see a Prius driving around, it must be a self-driving car, and therefore they can assume the right of way because they know the self-driving car will be cautious, and sometimes they'll be wrong.
That's a good point.
I'd like to bring up another issue of unintended consequences. When Mozambique was a colony of Portugal, which I assume is in the EU, the colonial administration mined most of the major cities and put large corridors about 2 kilometers wide with tens of thousands of mines around all the major cities. In 2000, there was a massive flood, and all these mines floated and got distributed randomly around the country. They're still there. And I mean, you think about it, the Adriatic is still full of mines, the Baltic is full of mines. These are problems that are going to be around for long, long periods of time. And I would think that the EU would get a little feel a little guilty about some of the residuals they've left about. You know?
If I can respond to that. So I am hearing great examples of robots that we all agree on and think are nice for a society. But if you look where funding is coming from, or if you look at, like, just pick up a magazine, what do people worry about? Well, it's about the robots, or maybe they're not robots, but at least they're drones that are used in warfare. And effectively, like a big amount of the money that we get is coming from that. I'm not saying that's bad or something, but I definitely think in discussing the ethics, we should be realistic. Like, where is our funding coming from, and where do people worry about? And there is a discussion that started. Like, there are several countries where people disagree that we use drones in war. Just to give one example, I think it's very complicated to do any research, and because if you're giving us example, right? If you can imagine that that it will be used in a way that's less nice for some members of our society, well, I think we can all imagine it for almost everything we've seen so far presented. That's my opinion. I'm not saying that we shouldn't do it, but it's definitely not a discussion for the future.
So I would like to switch topic now a little bit, because also at 7 we are going to close down this panel discussion. And I think there were a few topics put on the table that are actually important, in addition to the ones we have now been discussing, like ethics, implications, and so on. Like, some of you mentioned this whole idea of going for specific target systems and elaborate those in terms of machines, right? So Tim has a great interest in, let's say, more insect-oriented systems, Joe as well. Others might be more interested in vertebrate systems, like Tony. Yeah? So what I would like to hear now is, I do believe we can push the field forward by collectively targeting specific preparations. So it's okay, collectively we should really nail, let's say, Drosophila, okay, or we should really nail the elephant, okay. But so what are the opinions on these targets? What are the most promising target systems to, as a field, really make progress on this collection, these integrated sets of issues, right? So there's really like environment, body, and brain. So what are your opinions on that? What are these target systems we should really go for? And don't mention Angelina Jolie or so.
I wanted to comment because I think it injects one point that's important about the nature of our field. You know, Stefan was talking about the importance of basic research and the difficulty of getting funding in this area. In part, that stems from the fact that we're wearing, if we're doing this correctly, two hats. We're biologists and we're engineers. And one of the things that I was very proud of in arranging my own lab was that I managed to have under one roof the biology and the engineering. And I think what I've seen here at this meeting is that's the majority of the case here, that people are working with particular systems. That is a great challenge that every member of our community faces. Either you rely on the literature for the other side that you're interested in, and it doesn't have exactly the data that you need in order to accomplish your goal, or you do it all yourself. And I think that is a major issue for us to grapple with, is means to get funding agencies to acknowledge this field to support the necessity of having basically two research programs under one roof in some fashion. I hope that's a pertinent point.
I'd like to comment on the idea Paul put out of target systems, and I'm ambivalent about this, but my experience in the biological world has been some dismay at the promotion of a very, very few model systems at the expense of everything else, such that we now have the so-called genomic models in the US, which, you know, if you don't work on yeast, rats, Drosophila, zebrafish, or mice, forget it. You may as well not be, you know, in science, which is ridiculous. And the other side of that coin is that any model system is a model system because it is special. It is not like everything else, which brings up the problem of how generalizable is a target system. So the reason I say I'm ambivalent about this is because you could argue that the genomics explosion has been a huge boon to mankind and that perhaps that approach was the one to take. I might like to see if we can explore the idea of approaching problems rather than particular systems. if we all, and let me pick, if you all decide you're going to work on the caterpillar, which is what I do, great for me, right? I could probably get a lot of resources attracted to that model system, but pretty lousy for the guys working on octopus. So, but if we were to say there is a common problem, we are trying to understand how to control high deformation structures, then a lot of us can benefit from that regardless of the model system we work on. So I would like to argue, and perhaps that's what you're asking actually, that we work towards target problems rather than target systems themselves.
Can I just make a comment? I mean, I completely agree, and I think that the field of neuroscience has moved away from comparative studies in a significant way compared to maybe 20, 30 years ago when many people were doing comparative work. I mean, I've been doing a bit of comparative work ourselves, and you realize that just in rodent-like mammals we know virtually nothing about anything apart from mice and rats. Even mice and rats are very different, and we study whisking behavior, and people assume that a mouse and a rat whisk in the same way, but actually they don't. And they assume, therefore, that the cortex of the mouse and the rat is the same, which in fact it isn't. And so, but these differences are interesting, and we should be looking at them, because what are the differences and how do they relate to behavior? So we do need model systems because it's an enormous amount of work to start on anything new, but we need to look closely at differences between animals, because I think a lot of the secrets that the design principles are in the differences and the similarities.
And I would agree with Barry. I think what we do want to do is work on problems, and what I want to add to that is that the careful, and it goes along with your comparative comment as well, the careful selection of model species to work on the appropriate approach to the problem is a strength. So for example, we just ran this workshop on muscular hydrostats to develop soft robotics, a very desirable sort of thing. If we look across taxa, we see that there are muscular hydrostats in cephalopods, they're in lizards, they're in birds, they are in, and me right now, the tongue that I'm using is making speech possible. And so a wide host of possible applications unfolds if we tackle a problem area which we don't understand the technology. We don't understand our own technology for, and those are the appropriate kinds of directions, I think, to pursue with biomimetics, rather than reproducing things that are already in existing human technologies.
Yeah, well, I definitely agree again on the comparative aspect and how valuable it is, because we can isolate the general principles and the species-specific adaptations, and that's what we actually need. Maybe this is actually a community where comparative studies, for the sake of identifying general design principles, would be this would be a very good place to help funding these sorts of researches. And actually, in a way, it goes together anyway. So we identify a problem which we want to get solved in terms of robotic applications in the end. We identify the model organism that gives us some answers and some ideas. And if we really want to understand how it's done, then we, on top of that, we go for a comparative study. So I think that's the most complete or comprehensive approach one could go for. So I would say, do not, let's not focus on individual model systems which are fancy because you can knock out genes or something like that.
Any reactions to example systems?
Well, I think the focus on individual model systems was a constraint that was induced by the fact that sequencing organisms was very expensive. It's very cheap to sequence an organism right now, and I think that constraint is going to go away. So I don't see that as much as a limitation as it was 5 years ago.
Yeah, but Joe, you're not, for instance, if you take the Smetagessa ganglion, right, you go for the lobster. Right? You don't go for some other organism might have something equivalent, because in the lobster case you have the data, right?
Well,
so
we're sequencing that right now, and it's going to cost us about $5,000.
Sure.
Yeah, I mean it's not a big constraint anymore.
But I mean, in terms of the physiology, you have the physiology of the lobster, but not of many other species that might still have something like a Smetagessa ganglion.
That'll probably cost $300 in 5 years.
Yeah.
But the model system still is the one you have most information about.
Yeah.
Right. All right. Model systems. Anyone else wants to say something about model systems? Because then there's one more topic I would like to advance. Tim? Sorry? You want to add another model system? Don't say mermaids.
No, I think I agree with this point that to be so tied to a particular animal, I mean, and a particular species. There's this nice story when we started this AMOTH project that, so, okay, we got this project to translate chemical search from the moth, right? From the EU, they're all very excited, great, this is going to be fantastic, we love this. So we have our first meeting with the biologists and sit down, and so the question is, okay, which moth are we going to model, which are we going to study, right? Because there's about 50,000 of them, and they're the most numerous sort of phyla in the animal species, you know. So take your pick, right? So even when you sort of take a choice within that, so clearly the it's a balance between having canonical solutions and canonical models and mechanisms. So I don't feel as excited or as restricted as I used to at the beginning, when I really felt, oh, we have to really be faithful to every single detail in a particular animal. That I think that the mechanisms can largely be conserved, or the important mechanisms have some utility and portability across species if they're important, and this is nice because it means things maybe aren't quite as restrictive as we thought they were when we started, having to desperately latch on to one particular solution, consider nothing else.
Right. Okay. So then the other issue I would like to bring up is, I hope this works. So now, some of you mentioned, let's say, specific species you might want to model, but others mentioned specific applications you want to realize, right? So examples were demining, a caretaker robot, for instance. So also are there really, as a field, are there specific applications we should commit ourselves to? Or is there a range, a domain, an envelope of applications we should target in particular? Or to get mileage as a field, right? Or should that just be left open and we go for whatever looks interesting at the moment? Tim, are you still holding on to the microphone? Okay. So who has an answer? Joe, you have an answer.
Well, I think a capability is what I'm more interested in, and that's autonomy. Everybody here is kind of assuming autonomy's been solved. We're nowhere near it. Mm-hmm. You know, we can't even define it. I mean, I think that's a much more fundamental issue, is until we get a handle on what the problem is and get a path to solving it, all these applications are kind of a secondary issue.
Very good.
I don't think I'm going to necessarily suggest a single application, but I think that the thing that unites everybody doing biomimetics is that we're working towards devices that operate in the human world, if you will, and the natural world. And that's the key. A lot of us are saying, look, we don't have robots that can negotiate a city, and we don't have robots that can walk through a forest. They just don't have that capability, and that's what we're really trying to achieve, and then use that capability for our own ends to try and make life better for people. So what we're talking about is making machines that are safe to interact with humans and you know, natural environments. And I would like to add to that the potential of using biological materials so that we have green robots. I hate that, but it's kind of a catchphrase. Biodegradable robots, potentially. Certainly things that the world can use without us trashing the place, because I don't know if you've looked at car junkyards. They're pretty awful places because we have to get rid of that stuff that we've built. It would be really nice to compost your robot, wouldn't it? So it's just one idea, but I think that the thing that unites us is this sense that we need robots that are going to work with us in the real world.
Great.
So I think what we learn from biology is that most animals are specialists, and humans are an unusual animal in that we're extremely flexible and we can do all sorts of different tasks, largely because we've invested in a very complicated brain, which uses up a lot of energy. And in our field, in robotics, people have taken the human model, and there's a lot of work on that. And perhaps one of the benefits from looking at other model species is that we can see that animals solve difficult problems using a relatively limited investment in brain power. And some of them are solving difficult tasks, bypassing the challenge using morphology or materials or whatever. And if perhaps the lesson is not that there are specific applications, but that if we look for where we might use robots and where robots might be deployed successfully in the near future, it's probably on fairly specific tasks where a robot that is designed for that task will be able to do it well without having to have human level control.
I think the robot, I like more like such an assistive type of robot, or kind of such a supporting, human supporting type of robot. Maybe it's a little bit autonomous, but still listen to, intelligent enough to estimate the intention of the user. That's a kind of interesting. So even the wheelchair, even any other kind of master suits, those things, that's a kind of also interesting where to go. Don't go the zigzag, but they go straight, okay? That's a kind of just an intention estimation. That's a control problem estimation, those things. That's a very, also ethical problem too. So that's okay, good.
Applications, anyone? Preferences? David?
So I noticed that a lot of people are thinking about robots, but I think we should think broader than just robots. And not to mention, I don't want to mention Velcro actually, but I did like the Smith trigger example a lot, which I didn't think of straight away. I think there are many examples where people are actually interested in getting inspiration from nature for finding solutions for the design problems. And if you talk with companies in general, so they would be probably like the first to use our technology that we come up with. They are often more interested in the methodology that we use to come up with our solutions than the actual solution. It's something to keep in mind when you're developing a field, I think.
Excellent. Any reactions here in the audience? All right, then I thank everybody very much. That's great. So now you know where your future is heading. So let's see where this now is. I wanted to quickly inform you where the dinner will take place. But of course, I'm not on the screen yet. Let's solve that very quickly. So dinner is in a restaurant called Bo, or Boo. And, let me see, yep, here it is. Starting up. Well, that's not very useful, is it? Well, you can already see a red circle somewhere, but that is where we are right now. Okay, so let's kill that. I will restart this. Can you restart it, please? So the restaurant is at a beach called Marbella. It's in the area called Poblenou. There's a bus 41, that will bring you straight from about here to there. Now, as in direction, if you just go to the sea, so you walk to the sea, which is sort of east from here, and then if you walk east, you cannot miss the sea. Then essentially the restaurant is on a pier that sticks into the sea, and it's actually the only restaurant with that feature in the whole city. Okay, so in that sense you cannot miss it. We're getting closer here to the map. Give us the map. There it is. All right. So, so right now we're up here in A. Okay, and the restaurant is on this pier here. So this is in the wrong place. It's on this pier here. That's sticking out into the sea. It's Marbella. It's very close to a sports center that you cannot miss. All right, so it's very close to this pier. It's on the pier itself. It looks a bit like a ship on a pier. All right, so.
People get the metro to the Villa
Olympica. So you can basically take a metro to Villa Olympica, which is the area here, and if you then just walk along the beach, then you will hit it after a few hundred meters as well. It's another way to get there. Okay? And it's a really nice place, so please join us for dinner this evening, where we can then close the deal on our future. We meet at 8:30, to my knowledge. Anna, it's 8:30? Yeah. 8:30. All right.